Hello and welcome to this Nature Research custom media webcast titled Cryo-EM and Artificial Intelligence, a Marriage Made in Cell Extracts, exploring cellular organization in native cell extracts with high-resolution cryo-EM and artificial intelligence methods. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by GenScript. Protein interactions orchestrate a plethora of cellular processes and are thus essential to understand cellular function. However, their analysis is challenging, especially in the native context. Today, we're joined by Dr. Panagiotis Kastritis, Junior Professor at Martin Luther University, Halle-Wittenberg. In this webcast, he will explore the feasibility of deriving vital structural insights for protein interactions in endogenous cellular extracts that retain principles of cellular organization. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to Dr. Kastritis.
Okay, thanks really a lot for the invitation and the introduction. Indeed, we are going to talk about Cryo-EM and artificial intelligence, a marriage made in cell extracts. So actually, when we look inside the cell directly, like what Yulia Mahamid from EMBL Heidelberg did in 2016 with electron tomography, we can see a lot of dark densities around, and we can recognize some of them. For example, the nuclear pore complex, membranes, and ribosomes, and actin or tubulin filaments, as well as chromatin inside the nucleus. And you can understand that structural biology has progressed really a lot during the last years, where we can even see structures of complexes inside the cell. But as you can appreciate, we can see few of these structures. Traditional structural methods, they study our proteins and protein complexes in atomic detail, but they study them outside the cellular context, because very often we purify these complexes in homogeneity, and we lose what happens inside the cell. And here I said that you see a protein-protein interaction network from a cell, and all of these dots represent proteins, and their connections represent interactions, and you can appreciate this kind of complexity that we often miss when we study the proteins highly purified from heterologous systems. And what I continued saying without you listening to me, but what my lab is doing is basically to connect this kind of two structural investigation methods. So we are studying cell extracts that we are trying to bridge in situ structural biology and these traditional structural methods. And because now we are cell-free, our cell extracts are accessible to proteomic studies, cryo-electron microscopy investigations, and of course computational modeling. And we should not forget that cell extracts are expected to retain principles of cellular organization. Right? What do I mean by that? The genetic code, for example, was deciphered by using cell extracts, right? So using the tRNAs and having as readouts the amino acids that were being produced. Here are the first images that I acquired back in EMBL Heidelberg in 2017, where you can clearly see how cell extracts look under the electron microscope, and you can really appreciate this kind of higher order structures. And they form these higher order structures that Anne-Claude Gavin has coined the term protein communities, and now we start visualizing them, this kind of very complex pictures. And back then, I was able to resolve at 4.7 angstrom resolution the structure of this kind of hamburger-shaped molecule, the fatty acid synthase complex, mega-Dalton enzymatic machine. This kind of work set the notion that we can really start structurally characterizing what is inside our native cell extracts. And what we start seeing are these protein communities that I can show you here in this schematic, that basically enzymes or molecules involved in the same pathway, they organize together on top of membranes, scaffolds, or structural elements. They might form multimers and channel their intermediates from their product as a substrate from one enzyme, for example, to the other. And this contrasts how we are trying to connect our knowledge using traditional structural biology methods, where we have an enzyme or an enzymatic complex, and we are trying to understand its structure and function separately, and trying to connect the dots linearly. So this is another image that I acquired here in Halle, one of the first images from my electron microscopes in my lab, where you can directly see this kind of complexity. And this is a very beautiful image, negatively stained sample. And here you can see all of these kind of structural signatures that represent protein complexes and this kind of higher order assemblies. And by looking at such images, several questions arise. How do we discover and define protein communities and their members? How do we correlate protein community identity and protein structure? And eventually, how do we understand their activity? Because studying cell extracts and getting structures out of these is not that meaningful when you don't have function in your cell extracts, in order to start understanding how this function manifests in more complex systems. In order to answer these questions, I have devised in my laboratory a workflow that I am showing here, where we have model organisms, samples that we are using that come from cell extracts, either membrane complexes or soluble complexes, and then we apply our favorite methods, specifically cryo-electron microscopy, where in my laboratory we have our cryo-electron microscopes, mass spectrometry with collaborators, and computational structural biology using the Haddock software, for which I am a developer, and then a co-developer. And then by producing all of this data for the cell extracts with our methods, we include external database information, and we perform our analysis by integrating all of this data. And recently, as you will see in the presentation, we have incorporated machine learning approaches to understand better how our enzymes organize in complexes and higher order assemblies, and how these structures could look like. In order to do this kind of integrative work, of course, a lot of people are involved. Here I'm showing pictures of the people of my lab members. As you can see, we are an international lab, and also it is very important that people from different scientific backgrounds come together to produce the coherent structural data from such heterogeneous mixtures. Here I'm showing an image from cell extracts using cryo-electron microscopy, and again, with these dark densities, you can see a lot of different structural signatures, and I'm going to talk about this one here. So how do we know what is this, what is it doing, and what kind of assembly it forms? So, what we have done is basically we take our Hitomium thermophilum. This is a model organism from my lab, and it's a thermophilic eukaryotic mold, right? A thermophilic fungus that can grow to up to 55 degrees. So it's a very good model system because, as you know, thermophilic proteins and complexes are very stable, much more stable than their mesophilic counterparts. And then we perform our cell lysis and size exclusion chromatography of the native cell extract, collecting this kind of cellular fractions. And here you can see the size exclusion chromatography profiles that are highly reproducible that we collect. And you can see that what elutes here from the cell extract elutes according to their hydrodynamic radius, and in the first fractions we can enrich for actually megadalton complexes, like 10 megadalton down to 4 megadalton. And in this kind of fractions, we can see two specific structural signatures there. And when we do cryo-electron microscopy directly in this cell extract, we can see, for example, fatty acid synthase with a binder, or other kinds of very nice single particles. Here I'm showing you data like of 50 nanometer molecules of different kinds of beautiful signatures. And when we do large-scale analysis of images collected from this fraction, we can start seeing structural insights. And what do I mean by that? Is that this kind of signatures that we started collecting, they correspond to two different complexes. So to the pyruvate dehydrogenase complex, PDHC, and the oxoglutarate dehydrogenase complex, OGDHC. And these have basically molecular weights up to like 10 megadalton for PDHC and 4 megadalton for OGDHC. And here we have applied activity assays, and we see that these two complexes inside our cellular fractions, they are active. And also with mass spectrometry data, we can identify in these megadalton fractions all of their components. And even with Western blots and antibodies produced by GenScript, with which company we collaborate on getting this kind of antibodies, you can clearly see here that we can identify proteins that are assembling these megadalton complexes. In this case, the two proteins out of multiple proteins that organize the pyruvate dehydrogenase complex. And then what we do is that we start looking into these fractions with electron microscopy. And every cellular fraction, we do negative stain electron microscopy, and you can see the PDHC here in blue, the OGDHC complex here in red, collecting thousands of images in our electron microscopes. And then finally, when we extract these kind of single particles and do averaging, we can start seeing more and more clearly how these kind of two specific complexes are organized. And we can clearly see that they have this kind of cubic core for this kind of cubic complex, and this kind of symmetric core for this bigger complex, and are surrounded by several subunits. And then we can do basically 3D reconstructions of these particles and start seeing how they look like in 3D. And what you can see here is basically this kind of complexes directly from an endogenous cell extract, how the subunits are organized. And you can see again the reconstructions of the cubic core, the reconstruction of the icosahedral core, and the densities around that correspond to proteins that are involved in their enzymatic reactions that I have showed to you that these complexes are active. And we can capture these complexes in the different fractions, seeing this kind of densities around that often do this kind of clustering. As I said, this kind of core densities, cubic and icosahedral, they are surrounded by asymmetric densities that are not diffuse, and we have reproducible external densities that we are capturing. And for this kind of work, we wanted to look better and look with higher resolution what happens in this kind of icosahedral molecule, the PDHC. So in this work, what we did is again our favorite size exclusion chromatography, where we enrich for our 10 megadalton complex of the pyruvate dehydrogenase complex. Now with GenScript, we designed antibodies for all the subunits of the complex, and we can clearly see that all of them are together in the same fraction, meaning that we have the complex in our fraction. And we have developed in my lab better activity assays to probe actually reaction rates for our enzyme, which converts pyruvate to acetyl-CoA. Of course, like the PDHC, the pyruvate dehydrogenase, it's a textbook complex localized in the mitochondria. So here, after we performed this, we could resolve the structure of the core of the pyruvate dehydrogenase complex at 3.85 angstrom resolution, where we start seeing like residue-residue interactions. Here we can see the coenzyme A binding site that can occupy very nicely the coenzyme A, and we can start seeing densities for arginines, for example, forming an unusual pi electron network. What we can also start doing, because we have a higher resolution here at around 3.8 angstrom, is to start understanding how the mechanism in this kind of huge assembly works, and specifically at the core region. Here I'm showing a very simple schematic on the reaction mechanism of this extremely complex 10 megadalton assembly. And here it's a simple mechanism where basically the enzyme complex is composed of 3 subunits, E1, E2, and E3. Then you have the lipoyl domain, which acts as an arm that brings the substrate and the intermediate and the product from one enzymatic site to another, from the E1 to the E2 to the E3. And what we do is that here we started understanding the transcatylase reaction, where basically the coenzyme gets acetylated. And this kind of reaction happens in the core. What we see after doing the Cryo-EM reconstruction of our core and starting understanding with docking simulations how the lipoyl domain can bind to the E2 core. Here I'm showing a trimer extracted out of this icosahedral core. We can start understanding how the interface forms, what kind of energetics are being involved, and the buried surface areas, which are relatively low, and these indicate transient protein-protein interactions. And this kind of binding interface of the lipoyl domain with the E2 protein that forms the core, we can really see that its interactions are mediated by electrostatic complementarity, which is quite nice to understand, because this kind of electrostatic complementarity is very usual in this kind of transient complexes that need high on and off rates to interact. And here it's a view in the binding site at the E2 core, where you can see the lipoyl domain and the lipoyl lysine of this domain very nicely going inside the active site, and the coenzyme A, and how it is organized in a positively charged surface of the core. So, as you can see, from native cell extracts, we can go down to this kind of resolutions that we can start understanding how our enzymes might work in their kind, or in this kind of more native-like states, in this kind of heterogeneous samples. And what we see is not only to understand at higher and higher resolution this kind of reactions, but also understand the bigger picture, right? So this is the core where I described to you, and then on top of the core, in this kind of endogenous native pyruvate dehydrogenase complex, we see a crown-like density, which is basically very persistent. In our class averages, you can clearly see this kind of densities that are always in proximity to one another. And when we do the 3D reconstruction, we can see this kind of crown shape on top of our E2 core. And by doing a lot of calculations and docking calculations, benchmarking, we could unambiguously say and report that these densities correspond to the E1 enzyme, whereas this density corresponds to the E3 enzyme, whereas the other densities were ambiguous. But what we see also is this kind of density that surrounds our E1 and E3 proteins, confining them somehow on top of our E2 core. What does that mean is that basically right now we start understanding not the individual proteins that we already know their structure and function from X-ray crystallography and very advanced kinetics from the 50s and 60s and 70s, etc., but we can start understanding their communication. And here, in this kind of schematic, I am doing a schematic of what we have modeled here from our native cell extracts, the three-dimensional map. And we start understanding the distances between active sites, how the lipoyl domain that is bound by a flexible linker goes from one enzymatic site to another. Now we know we can characterize this kind of 3D architecture, and what we see is all of these observations that I'm listing here. This kind of reaction distances. We can see a reaction exclusion zone here with green, but also we see this prominent density that is formed by a cluster of E1 and E3 proteins around our more stable densities that correspond to the E1 and the E3 proteins. So, I talked to you about this complex right now, the pyruvate dehydrogenase complex, but what about the others, right? So, can we study all of these kind of complexes together? Now, with the advances in artificial intelligence, can we get more and more insights into their structures? So, again, here we are doing our very fundamental protocols that we have in my lab to separate with high resolution this kind of native cell extracts, collect the megadalton fractions and identify with mass spectrometry, activity assays, and Western blots with our GenScript antibodies where these protein complexes are and if they are having their subunits. And by picking all of these particles from the micrographs, we can start seeing different kinds of structural signatures corresponding to very minor abundances identified in the proteomic data. And what you can again see here is this interesting that these are like very stable cores, but they are surrounded by this kind of flexible diffused densities forming these higher order assemblies. And from these higher order assemblies, we could reconstruct their core components. And here you can see the core components that were reconstructed in 3D, completely de novo. And you can see a cubic density, an icosahedral density, this kind of hamburger-shaped density, and an asymmetric density. Here, with scale bars that are around 10 nanometers. And here we start identifying what they are. So what we do here is that we use this Omokage database from PDB Japan, and this tool compares our reconstructed densities to all the shapes that exist in the EMDB, in the electron microscopy data bank, and with those from the protein data bank. And for 3 of these, we can unambiguously characterize what they are, but for the 4th of these, because it was asymmetric, the algorithm could not detect it, but then by searching the hits, we could see from the 2D projections that we can identify also our 4th structural signature. By knowing now the symmetries, we can have higher resolution reconstructions, and here you can see very nicely some kind of resolved secondary structural features like helical bundles in the case of fatty acid synthase, in the case of this alpha-ketoacid dehydrogenase core, an intra-trimeric interface with the helices around, and even side chain resolution in the case of the core of the pyruvate dehydrogenase complex, and the rRNA pitch here in the case of the 360S ribosome. Here is where artificial intelligence kicks in. So what we did is something very interesting. So we have our native cell extracts unidentified map. So I told you that we have identified all the maps, but at what level? All the other maps here that I showed, we have identified exactly what kind of amino acid sequence fits. But for this cubic core, we didn't know, because the alpha-ketoacid dehydrogenase complexes, at least 3 of them have the same cubic core, so we didn't know this cubic core at this resolution of 4.5 angstroms to which one it corresponded to. So here we used AlphaFold in order to model the 2 enzymes that we find in our mass spectrometry data and are the candidates to fit in this density. Then we perform classical structural refinement of those and in our experimental map, and then we extract the backbones, and with the tool Find My Sequence, that is also an AI that is developed by Dr. Gregor Czonowski, our collaborator from EMBL Hamburg, we could basically understand which sequence matches with very high confidence in this kind of cubic map and which one didn't. And after we used AlphaFold and Find My Sequence AIs to understand which exact sequence fits there, then we could correctly identify what is there and build our final cubic 3D model. So these are the steps. As you can see here, we have our unidentified map. We do our model prediction with AlphaFold, and then with traditional tools, we are modeling the maps. Then we extract our backbones here from these maps, and using a second AI, we can understand what kind of sequence fits in our density and what kind of sequence doesn't. And then finally, we model our experimental model in our 4.5 angstrom map. Some insights here, you can see these beautiful structures. They are like all of the mega Dalton complexes from cell extracts. But with AlphaFold, we could see some secondary structures and that AlphaFold predicted that is actually not there in the experimental map. And it is very interesting because here, the backfolded N-terminal that you see here predicted by AlphaFold is absent in our experimental density, and this indicates that the endogenous state of the pyruvate dehydrogenase complex has much more flexibility. Here, also, AlphaFold could not model and see what is inside these additional densities, although the resolution was decent in order to fit somewhere there. And we have another manuscript where we discuss about these additional densities that basically correspond to an additional protein that we localized where it is inside the core. Then for the fatty acid synthase structure from AI, what we saw basically here after modeling it is that our hitomium structure from the native cell extract has some kind of conformational change in this acetyl-CoA binding domain here, and we think that it is mediated by this kind of flexible linker, that this flexible linker is again more flexible in our structure. And the AlphaFold model of this complex basically very nicely predicted the hexameric interface inside the core of the fatty acid synthase. As you can see here, with minimal refinement, we could recapitulate this kind of hexamerization interface in the core. But again, AlphaFold predicted this acetyl-CoA binding domain where the yeast model had it, and not where we see it in our experimental density around 15 degrees of displacement there. And finally, for the annotated now alpha-ketoacid dehydrogenase core, we can unambiguously say with our AIs which core it is. But before, again, I illustrate here the example, we couldn't know. So as you can see here, with traditional fitting of this kind of densities, we couldn't know if this core corresponded to the one enzyme or the other. But then with AI, we could not only understand this, but also understand something that, again, made an impression to us that, again, structural elements were predicted that basically were not there in the endogenous state of the protein complex. And here you can see the bound lipoyl domain. And this is extremely interesting structure that could partially exist transiently in our data, but our experimental data suggests that this density is absent, and if it is there, it's a very minor population. So overall, we are combining artificial intelligence like AlphaFold and this Find My Sequence by Dr. Tsonovsky, and also HADOC that I didn't cover in this presentation, that is developed by Alexander Bonvan in Utrecht, and I'm a co-developer of this program. And what we are trying to do is to understand how proteins interact, then by using experimental data, how they assemble into a bigger complex, and then how this bigger complex can be understood in the native cell extracts. And slowly to try to understand this kind of very, very complex interaction network, where by taking images of cell extracts using Cryo-EM, we start seeing one by one this kind of protein-protein interactions and networks, and start adding the visual component, how they are organized by disordered regions, by direct interfaces, these proteins and their assemblies. And by acquiring more and more data and applying our artificial intelligence methods that we are co-developing with EMBL Hamburg to add the structural dimension of the native context in this kind of interactons. So at this point, my presentation has come to an end. I would like to thank my lab members, my collaborators, of course, and my funding agencies, especially the BMBF, who is a major funder of my work and of my microscopes, my Cryo-EM microscopes, and also my other funders. And yeah, thank you.
Thank you very much, Dr. Kastridis. It is now time for the question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. Okay, so Dr. Kastridis, if I can ask you to stop sharing your screen, and that will bring us back into the studio. Our first question then for today: what are the advantages of studying protein structures in the native context of cell extracts as compared to, for example, in cell methods?
Yeah, of course. Yeah, this question, it was basically when the first time that my connection was broken, and I stated the advantage. So it's a very nice question, and it's part of the motivation that we are doing in the lab. So in-cell methods can see, of course, directly inside the cell, but the cell is so concentrated that, for example, by doing cryo-electron tomography of lamella sections, right now we are limited to seeing not more than 10 complexes in total. And in particular, for the Krebs cycle metabolomes, like the oxoglutarate dehydrogenase complex that I presented, the pyruvate dehydrogenase complex that I presented, inside the mitochondrion, if you cut the mitochondrion to see what is there, it's 400 milligrams per ml. And putting 400 milligrams per ml under the microscope, you just see all over densities. So the huge advantage is that you see these kind of complexes that are hidden in your when you see inside the cell. And also, of course, you increase your concentrations. So with lies like millions of cells, and therefore we have a biochemical sample that we can manipulate. We can enrich specific complexes. We can understand directly function. We can think of methods to probe like cancer cells or to probe a biotechnological product. So these are like the advantages of studying this kind of cell extracts compared to cells.
Thank you very much. Our next question asks, how can more higher order interactions be retained in the context of cell extracts?
Yeah, this is also another excellent question. What we are trying to do in my lab is like a lot of like even less harsh purification procedures or separation procedures of cell extracts. So the most like limiting step is the lysis. So when you lyse a cell, of course you dilute and you lose interactions. Now, how you can retain them? You can retain them, for example, as I said, by benchmarking milder and milder lysis protocols. But of course, it's very important to think of other methods like cross-linking them, either chemical cross-linking or photo cross-linking or whatever kind of cross-linking you can think of in order to retain this kind of transient interactions that might fall off during this kind of purification. But this comes in the expense of native cell extract and recovering possibly activities. So every time you have, again, advantages and disadvantages, and it depends on your scientific question. If you want to identify and know how your structure looks like with the transient binders, cross-link it. But if you want the more native state that you can capture with activity assays and KM values and everything and correlate it to structural data, then I would avoid it.
Thank you very much. Worth noting indeed. Our next question asks about disordered proteins. How is it possible to predict the potential conformations and or interactors of disordered proteins?
Yeah, you cannot right now predict the conformations of the disordered protein because it is disordered. But what you can actually predict is the ordered domains that this disordered protein binds to. For example, there are disordered proteins that bind other ordered proteins and bring them in proximity. And by knowing the length of the disordered region of the protein, then you can have physical chemical models, or like from the cross-linking data, have statistical models to know the proximities of your ordered domains. And this is what we did for the pyruvate dehydrogenase complex, which is full of disordered linkers that bring together ordered domains. What I showed of these proteins around the core were basically ordered proteins that were held by disordered linkers. So we cannot predict the structure of the disordered protein, but what we can do is infer the three-dimensional architecture of the ordered parts that surround and are organized by this kind of disordered regions.
Great, thank you very much. Our next question asks, what are the protein complex size limits for this kind of technique? Do they have to be in megadaltons in size, or will it work for smaller protein complexes?
Yeah, this is another excellent question. It's something that we are really trying to achieve also in the lab. Unfortunately, I didn't have 2 hours to present to you what we are doing here that I would like to, but for sure, size is a limitation, especially in cryo-electron microscopy and especially for complex mixtures. So we have a high-resolution structure of around 200 kilodalton complex. So I think that in 200 kilodalton we can go, but we really cannot go any further right now. But it also depends on the microscopes that you have, right? Here we have an older technology microscope, a geol microscope, the geol Gen 3200 FSC, and the newer technology microscope, the Glacios, but the Glacios is 200 kV. For example, if we had the Cryos G4 here, of course, we could even go to better and better signal-to-noise ratios. So I think that the limit could be like similar to single particle. This is what I think, around like 50 kilodalton, I would say 60.
Okay, thank you very much. Our next question asks, what the challenges are that are associated with studying protein-protein interactions in whole cell as compared to partial cell, so cells lysed with some sort of detergent?
I do not understand the question. Can you repeat?
Let me repeat it to you. What are the challenges associated with studying protein-protein interaction in whole cells as compared to partial cells? And the example was given a cell.
Yeah, I'm not a cell biologist, so I'm a biochemist and structural biologist, so I cannot answer exactly and in detail what are the challenges of studying protein-protein interactions inside cells. What I know is that, for example, if you add a fluorescent probe in your protein, of course, then your protein is not the same. So this is by itself a challenge. Now, other challenges I cannot say about what is happening in cell, but I can say the challenges after you lyse the cell. Of course, you have, after you lyse the cell, like us biochemists do, a lot of unspecific interactions that you have to understand which ones are unspecific and which ones are specific. So with different lysis protocols, you have readouts of the protein-protein interactions and if they are like reproducibly identified or not. But eventually, the protein-protein interactions that you capture, they have to be, in my opinion, validated by activity and function. So if you have, for example, function of the production of acetyl-CoA, and you have from your mass spec the metabolic pathway of producing acetyl-CoA, then you know that this kind of metabolic pathway is active there. So you know that this kind of protein-protein interactions are real because you have your product, you have a function. So this is how we perceive ourselves discovering protein-protein interactions that are functionally relevant. Of course, like we can have a lot of prediction models from String, from BioGrid, from all of our amazing databases with like different methods like in-cell cross-linking and photo cross-linking and systematically identifying binders using immunoprecipitation mass spectrometry and all of the other beautiful methods. But in my opinion, like probing activity is extremely important to have high-quality protein-protein interactions that are functional.
Yes, and actually on that, there's a further question which is, asking whether it is better to do cross-linking prior to lysis or after lysis. But what you're saying is after.
I think after, if you want to retain interactions that are reproducibly identified. You can also do, of course, before, because then you can capture what is happening in cell, but then you don't know what you are capturing is active. So I would make an approach where I am studying without cross-linking, then I'm cross-linking the fraction, then I'm cross-linking the extract, and then I'm cross-linking the cell and trying to connect all of this kind of data to have a coherent functional metabolic pathway model.
Okay, thank you very much. Next question asks if this will work for mammalian cell extracts.
Yes, not only for mammalian cell extracts, but actually we have very beautiful data that unfortunately I cannot share because they are proprietary data. But what I want to tell you is that we are doing this same method for mitochondria of a specific cancer cell line, and we can probe how an enzyme can change in this kind of cancer cell line. So yes, it can work. The resolution of the gel filtration will not be the same because the thermophilic fungus has amazing advantages to study it. We should not forget that the ribosome structure from Adayonath came from thermophilic ribosomes, or like the proteasome from Baumeister group came from a thermophilic source, right? So thermophilic molecules are extremely advantageous to discover things. But yes, it works for mammalian cells, and especially the mitochondria.
Excellent. Thank you. Our next question asks, do you have a system or method that can monitor interactions between known interactors in a time-dependent manner?
Ourselves, we don't. There are a lot of methods that are doing it about known interactors, but you have to somehow always chase them with labeling, so to see them, right? So like Fred, you do this, or any kind of method, and in cell extracts, you can use different methods by labeling them. Like you can even use do NMR to probe like interaction in dynamics at room temperatures, and by having them heavy labeled, you can see these things. But it's not something that we are searching right now in my lab to do it in a time-dependent manner. What we do is the activity assays, for example, that are of course time-dependent. We monitor with our plate readers when the reaction happens, and some of these reactions, they are in the regime of also like freezing them for Cryo-EM and seeing the structures. So the time dependency for us is this, but of course you get average structures, you have other problems there. So the short answer to this question, we would really like to have it.
Okay, thank you very much for that. Our next question then, are you aware of any methods to expand your approach to the soluble cell extracts, also towards membrane proteins?
Yes, this is another excellent question. We all know that membrane proteins are extremely important, like they are like the majority of our pharmacological targets. And this is exactly what I'm doing with here in my laboratory. We are trying to do like membrane extracts. So the methods that we use for this that are somehow successful, but we are not there yet to have like this kind of extreme novelty, right? Because we have a lot of novelties, we don't have there the extreme novelty. We use like nanodiscs in order to solubilize our native membrane. So what we do is that we take our membranes of the fungus by ultracentrifugation, and then we apply our polymer there. So we use polymer nanodiscs to solubilize the membranes and get 10, 15 nanometers nanodiscs. And this is extremely important to use polymer nanodiscs because they are unique in the sense that you don't use at any step detergent, so you can extract directly your complexes from the membrane and see how they look like. Right now, as I said, we have very good results from that, but we don't have yet an atomic resolution structure to show it in my presentation. So yes, there are a lot of methods that we are developing in the lab for that, and my direction of my lab is towards polymer nanodiscs and their application.
Okay, thank you very much for that. Our next question, what do you think is the future of studying cell extracts besides the integration with artificial intelligence methods as you've shown today?
Yeah, so I think that the future of this method is extremely bright in the sense that we can start thinking of like prototyping a lot of our observations. For example, we can go towards biotechnology, right? We can start discovering in the cell extracts with activity assays new functions. We can study any kind of organism in an integrative model and their most abundant proteins, like this kind of exotic anamox bacteria, for example, to see like how all of these things come together in the cell extracts, and finally go to a direction of biotechnology to start having and understanding deeply the products and how to increase yields, to manipulate them, these kind of things. Membranes that we discussed, and membrane complexes, and higher order membrane assemblies, this is also one of the future, because if you start understanding them in their endogenous state inside cell extracts, then you can start testing drugs, testing activities, and in a more native environment, and by having readout structures. And of course, what EMBL Heidelberg is doing, that people should go out of the labs and to the nature. So EMBL has this kind of Tara Oceans expedition. Imagine that you take water from the nice sea of Greece, right? And you start looking what kind of organisms are there, they cell extracts, and start identifying and making libraries of these beautiful structures. So to understand basically better our world. So there are a lot of things that I can say, but due to the lack of time, I can also answer later with writing an essay. Yeah.
Okay, well, thank you very much. And on that note, we are going to have to finish for today. We are aware that there are some questions that we didn't manage to get to, but we will get back to you offline using the details that you've provided after the broadcast finishes. So I would like to thank Dr. Panagiotis Kastritis once again for his presentation today and for answering your questions. I would also like to thank the webcast sponsor GenScript and you, the audience, for tuning in to this webcast today. Remember, you can watch this again at any time on demand at nature.com/webcasts. Thanks for joining us, and I hope we'll see you again soon.